This is Biology, Chapter 11, Section 2, Complex Patterns of Inheritance, and our main idea for this section is complex inheritance of traits does not follow inheritance patterns described by Mendel. The essential questions for this section are: what are the differences between various complex inheritance patterns? How can sex-linked inheritance patterns be analyzed? And how can the environment influence the phenotype of an organism? So here we have incomplete dominance, which means dominant fails to be complete. In some organisms, heterozygous individuals will display the dominant phenotype. That's how we've been explaining it so far. However, with incomplete dominance, the heterozygous phenotype is an intermediate phenotype between the two homozygous phenotypes. Well, what does that mean? That means here we have red, which is homozygous dominant. We have white, which is homozygous white. If we think back to Mendel and his pea plants, the result, the offspring, if you will, would have been red, because R, with the tall, always dominates the small. However, in incomplete dominance, you get something more of in the middle. So a mix of red and white equals pink. So both the dominant and recessive alleles are expressed in heterozygous individuals or hybrids. Codominance, that's a little bit different. Codominance, let's look at sickle cell disease. It changes the hemoglobin that causes red blood cells to become sickle-shaped. People who are heterozygous for the trait have both normal and sickle-celled shapes. That means that they're both living together in a co-living situation. Sickle cell disease and malaria. Those who are heterozygous for the sickle cell trait also have a higher resistance to malaria. The death rate due to malaria is lower, where sickle cell trait is higher, meaning more people live to pass it on to their offspring.
So today we're going to talk about codominance and incomplete dominance. But first, let's review the example of blood type, and how someone with the same two alleles coding for the same trait would be called homozygous, and someone with different alleles would be called heterozygous. Also remember the concept of dominant and recessive alleles, and how the A allele is dominant over the O allele in this example. This means that the same phenotype, blood type A, can result from these two different genotypes. Now, the example that I just gave you was an example of complete dominance. So if the person had a genotype AO, since our phenotype is just blood type A, it means that the A allele is completely dominant over the O allele, and only the A allele from the genotype is expressed in the phenotype. But there are actually three different patterns of dominance that I want you to be familiar with. And to explain this, I'm going to use a different example. So let's say we have this flower, and the red petal phenotype is coded for by the red R allele, and the blue flower phenotype is coded for by the blue R allele. So I'm going to introduce three different patterns of dominance, and they are complete dominance, which you've already heard of, codominance, and also incomplete dominance. And I'm going to explain what these two new patterns are through this flower example. So let's start by looking at three different genotypes and the phenotypes that you would see for each of them under each different dominance pattern. So we'll start with the genotype 2 red Rs, which we can expect that in all cases the flower petals will be red, since we only have red Rs in the genotype. Similarly, if our genotype had 2 blue Rs, then we can expect that in all cases the flower petals will be blue, since we only have blue Rs in the genotype. Now these three different dominance patterns change when we look at the heterozygous example, and that's what makes these three patterns different. Now, we're already familiar with the example of complete dominance. So if we said that the red R is dominant over the blue R, then this would make the heterozygous phenotype a red flower for complete dominance. Now, what codominance is when the heterozygous phenotype shows a flower with some red petals and some blue petals. So it's when the two alleles are dominant together. They are codominant, and traits of both alleles show up in the phenotype. Now what incomplete dominance is when the heterozygous phenotype shows a mixture of the two alleles. So in this case, the red and blue flower petals may combine to form a purple flower. Neither allele is completely dominant over the other, and instead, the two, being incompletely dominant, mix together. So what did we learn? Well, if we assume the heterozygous genotype, red R, blue R, then there are 3 different dominance patterns that we might see for a specific trait. In complete dominance, only one allele in the genotype, the dominant allele, is seen in the phenotype. And this was the example with the red flower. In codominance, both alleles in the genotype are seen in the phenotype. And this was the example with the flower with both red and blue petals. And finally, in incomplete dominance, a mixture of the alleles in the genotype is seen in the phenotype. And this was the example with the purple flower.
And this time we're going to be looking at incomplete and codominance. The first one here we've got is incomplete dominance. And in incomplete dominance, 2 traits are not completely dominant over each other. In the past, in the original Punnett square problems we've dealt with, you've got capital T, which is dominant over the lowercase t. That's traditional dominance. Incomplete dominance, neither one of them is dominant, and so we're going to use this scheme here. We're going to use one set of letters for one trait, and another set of letters for the other trait. Now you're going to see this done differently in some books. Some books will have red flowers being this, and then white flowers being this with an apostrophe. It's red R prime, R prime. Not too crazy about that system. So I like this one a little better. So that's what I want to show you. So capital R would be red flowers. W would be white flowers. But when we have the R and the W together, the red and the white blend in the heterozygous. So we have pink flowers when we have R and W together. So let's look at how that would work in problem form. In certain species of fish, blue scales are dominant to yellow scales. Blue scales and yellow scales are incompletely dominant. Predict the outcome between 2 heterozygous fish. Well, in this kind of problem, the first thing I'd recommend you doing is making a key. And so I'm just going to take the very first thing it says, blue scales, and I'm going to make this blue. The next thing says yellow scales, so I'm going to just use Y. And then BY would be my heterozygous, and yellow and blue, when they mix together, make green.
You need to be writing this down in your notes off to the side or at a piece of paper.
So it says predict the outcome between 2 heterozygous fish. Now 2 heterozygous fish is going to be this one. So these are 2 green fish that we're going to be crossing. I'm going to make my Punnett square. 1 letter over every box. And then I'm going to fill it in just like we have in the past. I'm going to come in and do phenotype of, tell you what I'm going to do. I'm going to do genotype first. The genotype, I've got 1 that looks like this. I've got 2 that look like that. I've got 1 that looks like this. Now we come down to phenotype. This one, according to our key, is blue. So 25% of them is going to be blue. 2 of them are BY, which our key tells us is going to be green. 1 of them is YY, which our key tells us is going to be yellow. So the interesting thing here is that both the genotype and the phenotype are going to be the same percentages, 25, 50, 25, 25, 50, 25. And you're going to find that to be the case in these problems. Alright, the next kind of unusual thing, I always tell my students that once we learn the basics of genetics, so like in English, when you learn the basics of English, then you have to learn the exceptions. So here, this one, incomplete dominance, and this next one are the exceptions to Mendel's laws, kind of. Alright, this one's called codominance. In incomplete dominance, neither trait is dominant over the other. In codominance, the 2 traits are both equally dominant. And I know that sounds a lot alike. Just make sure you read the problem and look for the words. codominant and incomplete dominance in the problem. So we're going to use a little bit different, I'll change this right quick. A little bit different key here, but it works basically the same way. FB, FB are black feathers. FW, FW are white feathers. FB, FW are black and white spotted or speckled, which is kind of important. Spotted or speckled for the heterozygous. Now you're going to see this kind of format again when you get your blood type, because blood type is also codominant. So let's look at a problem using these. In some species of frogs, green skin and purple skin are codominant. Predict the outcome between two heterozygous frogs. Once again, first thing you need to do is make your key. So I'm going to start off by doing, since we're dealing with frogs, I'm going to use F, and then green skin, I'm just going to use a G.
Again, make sure you're writing this down.
So F, G is going to be green skin, and then these frogs have purple skin. So, and then we'll need a frog that has one green allele and one purple allele for the heterozygous. So this one would be green, this one would be purple, and this one would be, remember, we're just going to call it spotted. It'll be green and purple spotted skin. So the question asks about two heterozygous frogs. Alright, we're going to use this one, because that's heterozygous, and you're just going to fill it out just like you do your regular Punnett squares. FP, FG, FP. Except when you do something like that. It's okay, mistakes are part of life. G, FG, FG, FP, FG, FP, FP, FP. Alright, we're going to do our genotype again and our phenotype. I got 25%. That looks like this. I've got 50%, these two here in diagonal. Our FG, FP. And then that last one in the little corner down there, FG, excuse me, FP, FP, FP. Alright. So down here in the phenotype, this one, according to our key, is going to be green. These two in the middle, according to our key, are going to be spotted. And this last one in the corner, PP, according to our key, is going to be purple. Alright, and that's how you do codominant and incomplete dominance. Remember, in incomplete dominance, you're going to get a blending. In codominance, you're going to get spotted or speckled. Alright?
Continuing on, multiple alleles, blood groups in humans. Some forms of inheritance are determined by more than two alleles, referring to as multiple alleles. The ABO blood group has 3 forms of alleles, sometimes called AB markers. Stop now and go to part 2.